In the last lecture presentation, we discussed the innate immunity and the first and second lines of defense. The innate immune system is also referred to as our non-specific immune system, as its mechanisms offer general immunity to a wide variety of invading pathogens. The first line of defense was our body's skin, mucous membranes, and its secretions, and our body's second line of defense included phagocytes, natural killer cells, inflammation, antimicrobial chemicals, including interferon and complement, and fever. All of these mechanisms that we've discussed so far generally protect the body against foreign substances. However, nothing in this innate immune system is specific, and the innate immune system also does not display memory. So it is here with the adaptive immune system that we're going to start seeing specificity and memory being displayed. The adaptive immune system includes our body's third line of defense, which can be described as our immune response to specific antigens. If for some reason a pathogen is able to make it past our first and second line of defense, they will then have to jump the hurdle of antigen-specific defenses. So you can see on the right side of this slide our innate and adaptive immune systems broken down into their components. Remember, the innate immune system composed our first and second lines of defense. The surface barriers were the first line of defense, and the internal defenses is our second line of defense. The individual components that make up each line of defense are listed below. So this slide should serve as a little bit of organization for our immune system so far. But this picture goes a little further to also show us that we're going to be able to break down our adaptive immune system also. So let's first distinguish the characteristics of the adaptive immune system as a whole, and then we'll start to break down its major branches. So first and foremost, as I've said several times, the adaptive immune system displays both specificity and memory. Those are the key system regarding the adaptive immune system as a whole. Instead of offering general protection, if a particular pathogen is making its way into our body and causing us additional strain, certain immune cells can be employed to grab onto the individual antigens from those cells, process them, and then produce receptors that are specific for that antigen individually. Therefore, when that pathogen tries to make its way back into our body, our immune system is primed with receptors for that antigen before it even gets there. We'll also see that memory is another characteristic that the adaptive immune system displays that our innate immune system does not. Once our immune cells are triumphant over a particular pathogen, many of the cells that were used to fend off that pathogen will eventually break down. However, not all of the cells that were produced to defend against that pathogen will disappear. We will have certain memory cells that will remain after our adaptive immune system is stimulated. These memory cells can exist for decades and be ready to go whenever that pathogen tries to make its way back into the body. Immunological memory is the factor that makes the first exposure to a particular pathogen the worst. Every subsequent exposure to that exact same pathogen will produce a stronger, more powerful immune response, so you generally don't experience as bad symptoms the second time around. Our adaptive immune system also tends to act more systemically. With the cells and factors that are produced with the adaptive immune system, they tend to travel throughout the blood and are not restricted to the localized infection site. We will find two branches of our body's adaptive immune system, the humoral immune system and the cell-mediated immune system. The cell-mediated immune system includes our T cells, also known as T lymphocytes. They come from the lymphoid cell lineage. And our body's humoral immunity includes B cells, which are also known as B lymphocytes that also come from the lymphoid cell lineage. T cells, B cells, and NK cells are the three cells that do come from the lymphoid cell lineage, so we can refer to those cells as lymphocytes. Lymphocytes were the second most common white blood cell that our body had, and it will be the lymphocyte that will direct our entire adaptive immune system. So this slide will tell you much information that I've already in fact told you several times. The adaptive immune system displays both specificity and memory, and it has two major branches, our cell-mediated immunity and our antibody-mediated immunity. Antibody-mediated immunity is just another word to say our humoral immunity, which includes our B cells. We call this the humoral immunity because antibodies exist within our body's humors, such as interstitial fluid and blood. The cell-mediated immune system, on the other hand, is directed by our T cells, and both T cells and B cells will mature in different places. The B cells will mature in the bone marrow, which is where the rest of our blood cells are going to be formed at. The hemocytoblast that exists inside of our body's red bone marrow will produce the B cells, and they'll mature in the bone marrow. When they are released from the bone marrow, they are ready to act as members of the immune system. However, the T cells require a little bit more for their education to take place. T cells will be produced in the bone marrow. However, they will not mature until they make their way over to the thymus. T cells are going to be released from the bone marrow as immature T cells. They will migrate to the thymus, and thanks to thymosin and thymopoietin, our two major hormones that come from the thymus, the T cells will mature here and eventually be released from the thymus as mature T cells. We will soon break down our body's cell-mediated immunity and our humoral immunity, but we are first going to talk a little bit more about antigens, as they are the players that direct this entire branch of the immune system. Remember, antigens are cell-specific. Each cell has its own specific and unique antigens. So by recognizing the antigens on the surface of invading pathogens, our immune system can learn to attack specific individuals that could be particularly problematic. Antigens get their name from their ability to stimulate the immune response. They are, in fact, antibody generators. And this is where we get the word antigen from. So we're first going to talk a little bit more about antigens and antibodies as a whole, and then we're going to relate those back to our cell-mediated branch of the adaptive immune system and our humoral branch of the adaptive immune system. So far, we've discussed antigens as proteins that can be found on the surface of foreign substances. But antigens are not foreign by definition. There are also antigens that our own cells will have that our bodies can recognize as harmless. Those are termed self-antigens. Let's think back to blood typing to give us a little bit of insight about self-antigens versus non-self-antigens. If you are an A-type individual, meaning that your blood type is A, that means that you're going to have A antigens on the surface of your red blood cells, and therefore you'll have B antibodies in your blood also. So a non-self antigen for you, if you are an A-type individual, would be the B antigen. You don't naturally have the B antigen on the surface of your red blood cells because you're an A individual and you have the B antibodies. So therefore, the B antigen would be a non-self antigen. But you have the A antigen on the surface of your red blood cells if you're an A-type individual. Therefore, the A antigen will be a self-antigen, and it will not stimulate an immune response. All of that is reversed for a type B individual. If your blood type is type B, the B antigen will be your self-antigen, and the A antigen will be your non-self-antigen. It is here that we can hopefully understand that all of the antigens that are unique to you individually are your own self-antigens, and all of my antigens would be considered my self-antigens. However, your self-antigens would be non-self-antigens to me, and my self-antigens would be non-self-antigens to you. Since everyone has their own unique individual antigens, the phrase self-antigens is relative based on the person. One of the human's primary self-antigens is the major histocompatibility complex. The major histocompatibility complex proteins, also known as MHC or HLA, which stands for human leukocyte antigens, are one of the human's primary self-antigens. As I just explained, there are different types of MHC proteins, and different people will have different versions of this protein. Our white blood cells will patrol the body looking for our own MHC proteins. And if it encounters any other different types of MHC, that's going to stimulate the immune response. This is extremely relevant to transplantation. As I just mentioned, your self-antigens would be foreign antigens to me, and my self-antigens would be foreign antigens to you. Therefore, with transplantation, when we take an organ from one person and try to put it in another, we have to be absolutely sure that the MHC proteins are matching. We will talk more about transplantation towards the end of this chapter. However, we first must break down the difference between complete antigens and incomplete antigens. Antigens are in fact quite large molecules. You can see that this is representing an antigen here. And each antigen is going to have tiny little groups that are sticking off of the sides, known as antigenic determinants, or also called epitopes. It is these epitopes that are going to stimulate the immune response. The two functional properties of antigens as a whole are immunogenicity and reactivity. Complete antigens will display both immunogenicity and reactivity, and incomplete antigens, as we will come to see shortly, will only display the characteristic of reactivity. So let's break down the difference between these two functional properties. Immunogenicity is the ability to provoke an immune response by stimulating the production of specific antibodies, the proliferation of T cells, or both. In other words, immunogenicity refers to an antigen's ability to provoke an immune response. Reactivity is a little bit different. Reactivity is defined as the ability of the antigen to react specifically with the antibodies or cells that it provoked. In other words, reactivity defines the ability of an antigen to bind to antibodies or perhaps to stimulate lymphocytes. Reactivity is the ability for an antigen to bind, while immunogenicity is the ability of an antigen to bind and provoke an immune response. When we use the word antigen, we assume we're talking about complete antigens that display both reactivity and immunogenicity. However, incomplete antigens are going to be a little bit different. Incomplete antigens will display reactivity, but they don't display immunogenicity. So therefore, they can bind to antibodies or lymphocytes, but they can't provoke an immune response. Haptens are an example of incomplete antigens. Haptens can only stimulate immune responses if they're attached to larger carrier molecules. In order for a hapten to start displaying immunogenicity, one of our own proteins needs to bind to it, and that will help it start to provoke immune responses. One example of a hapten is poison ivy. Poison ivy releases a toxin that, after coming in contact with a carrier protein, will start to provoke an immune response. Peptides and nucleic acids can act as incomplete antigens. However, proteins, carbohydrates, lipids, and nucleic acids can all serve as complete antigens. While all of these can act as antigens, proteins tend to be the best substances to serve as antigens. Let's bring ourselves back to the antigenic determinants. Proteins will have little tiny groups that are sticking off the sides, and our antibodies can bind to those individual epitopes that are found on the antigen. Since proteins are so unique between different organisms, proteins will have many antigenic determinants that we can use to create antibodies. One other quick side note is that plastics are not very good antigenic determinants. Since plastics are molecules that have a lot of repeating units within them, there isn't a lot of variety or epitopes with plastics. So plastics often have the ability to be used for different procedures. Two examples of these are artificial heart valves and joints. Since plastics do not stimulate the immune response, even though they're a foreign substance, we can use them to reinforce joints or also to improve the structure of artificial heart valves. So now that we've discussed what antigens are and their different types, we are ready to start looking into the process as to how antigens stimulate the immune system. Remember, the adaptive immune system includes both the cell-mediated immune system, which is facilitated by our T cells, and our humoral immune system, which is facilitated by our B cells. We will first discuss the cell-mediated immunity and T cells. Now, T cells are very particular cells. They don't like to get their hands dirty. So in order for T cell activation to occur, another type of cell, known as an APC, an antigen-presenting cell, must process an antigen and present it out to the T cells. The three cells that can act as antigen-presenting cells are the dendritic cells, macrophages, and B cells. These three cells will undergo the process shown here at the right. They will phagocytize a particular antigen, break it down and process it, and then display the little pieces of that antigen on the surface of their cell membrane. These antigen-presenting cells will then come in contact with T cells, and through a series of receptor interactions will stimulate T cells to create a T cell receptor that is specific for that antigen. So let's take a look at this process. First and foremost, some type of antigen needs to come in contact with the cell. The cell will of course phagocytize that particle into a phagosome and eventually break down the contents of that phagosome with digestive enzymes. All the while this is going on, we have the synthesis of MHC2 molecules. Remember, the major histocompatibility complex proteins are one of the most basic self-antigens. So therefore, we're going to produce these MHC proteins so that the T cell that comes in contact with the antigen that we're about to present will also be able to understand that it's one of our own cells that's presenting that antigen instead of a foreign substance that's coming along by itself. Once the MHC2 molecules are produced, they're going to be released in a vesicle and eventually fuse with the vesicle that contained the endocytosed material. The antigen will bind to the MHC2 protein, and that will eventually be exocytosed onto the cell membrane. So now we have an antigen-presenting cell with an MHC2 receptor and a processed antigen that is attached. You can see the key here shows you which one is MHC2 and which one is the antigen. This antigen-MHC complex is embedded in the membrane and is the basis for antigen presentation. So again, just to be clear, our T cells aren't able to do this process on their own. They have to have antigens presented to them. But once the T cell gets an antigen presented, it can start to mediate the immune response and produce other types of T cells that will be able to go out and destroy any similar antigens that are displayed on the cell membrane of any invading particles. So these three cells will be our antigen-presenting cells, the three cells that can undergo this process. They will take in some type of antigen from the external environment, break it down, and put it on the MHC2 proteins, which will then be displayed on their membrane, so they'll be able to go and interact with T cells. So this slide shows us an overall picture of the adaptive immune system and its two branches. We know that the red bone marrow is going to be producing B cells and T cells, and it does produce mature B cells on its own. However, immature T cells will leave the red bone marrow and make their way over to the thymus, where they will become mature T cells. Our mature T cells, of which there are several types, will eventually be activated by antigen presentation. Once T cells are activated, they undergo a process known as clonal selection. Clonal selection is the process by which T cells will proliferate and produce many different clones of themselves. By increasing the number of T cells, we're better equipped to fend off pathogens with that specific antigen that stimulated clonal selection in the first place. We will shortly see the difference between cytotoxic T cells and helper T cells, but essentially both of these types of cells will undergo clonal selection to increase their numbers so that we're better equipped to fend off infections. Differentiation is also a very important player in clonal selection. Differentiation indicates that these cells undergoing clonal selection will change forms and produce different types of cells. Of course, we'll produce active versions of our cell in general, but we'll also be producing memory cells as well, and that requires a differentiation. B cells, once activated, will also undergo the process of clonal selection. They will differentiate into plasma cells and memory B cells. We'll talk about these a little bit more when we get to the humoral immunity. But first and foremost, we need to discuss the cell-mediated immunity and what happens when T cells are activated. So the first thing that I need to mention about the cell-mediated immunity is that we have three different types of T cells: helper T cells, memory T cells, and cytotoxic T cells, also known as killer T cells. Helper T cells are often referred to as CD4 cells because they have a CD4 protein that sticks off the side of their cell membrane. You can see that this is the T cell and this is the antigen-presenting cell. We already know that the MHC2 proteins will be displayed on the surface of our antigen-presenting cell with the antigen bound to it. This complex is going to be used to activate our T cells. So here you'll see our T cell receptor that will eventually bind to the antigen, and the CD4 protein will come around here and bind to the MHC2 protein. This is precisely why we call our helper T cells CD4 cells. Once an antigen-presenting cell with an antigen displayed on its cell surface comes and locks in to an inactive helper T cell, the binding of the T cell to the antigen and the CD4 protein to the MHC2 molecule is what will lead to activation of that helper T cell. Once activated, helper T cells will undergo the process of clonal selection, which is the proliferation and differentiation into many different clones. You'll see that we'll produce many different types of active helper T cells. These are clones of the original activated helper T cell. These clones are going to be used to help fend off the infection that our body's undergoing. But we'll also see that there's a differentiation into memory helper T cells also. These memory cells are the ones that will remain in the body for decades to come. Our active helper T cells will eventually die and degrade once the infection is at bay. But the memory helper T cells will continue to exist within the body so that they're ready and on scene if that antigen ever shows up again. Now, helper T cells in general, on top of needing antigen presentation, still don't like to get their hands dirty. These are the coordinators of the immune response. They will primarily be releasing different factors that will stimulate other cells to come into that area and fight that pathogen off. Helper T cells will also release a chemical known as cytokines. These will stimulate the growth of other T and B cells and continue to stimulate other cells to come to that area. We also have our memory T cells that will continue to stick around after the infection in case that pathogen makes its way back into our body. But one thing that we need to note about the memory T cells is that we can have both memory helper T cells and we can have memory cytotoxic T cells. Memory cytotoxic T cells act just as memory helper T cells do. They lie in wait in the body and wait for that antigen to surface once again. But cytotoxic T cells work a lot differently than the helper T cells do, and that's why these cells get the name killer T cells. Cytotoxic T cells are also referred to as CD8 cells because they have a protein on their cell membrane known as the CD8 protein. The CD8 protein will bind to a different type of MHC protein known as MHC1. But you can see virtually the same complex is shown here with activation of cytotoxic T cells. This MHC protein will be displaying the antigen, our T cell receptor will be bound to the antigen, and the CD8 protein will bind over here to the MHC protein on the surface of an infected body cell. There are a couple different types of MHC, and particularly cytotoxic T cells are stimulated by what's known as the MHC1 protein. When one of our body cells is infected by some type of microbe, that cell is going to do whatever it can to destroy that substance inside of it and display the antigens on its surface. And for infected body cells, this is where the cytotoxic T cells will come into play. We call these CD8 cells cytotoxic because they're toxic to our own cells. They're going to be prepared to destroy any type of our own body cells that have been infected by something else. By doing this, we can fend off the microbes that have tried to make their way into our own body cells. So once an infected body cell has locked into an inactive cytotoxic T cell, that's going to eventually lead to the activation of the cytotoxic T cell, which will then lead to its clonal selection, proliferation, and differentiation into active cytotoxic T cells and memory cytotoxic T cells. This is essentially the same exact process of helper T cell activation. Cytotoxic T cells get their name because they're more of a fan of hand-to-hand combat. These cells are going to make their way out, grab onto the infected body cell, and insert perforins in the cell membrane that will poke a hole and lead to cell lysis. Cytotoxic T cells are especially helpful with virus and cancer cells, as virally infected and cancerous cells both have in common that they are not a foreign cell altogether. They're one of our own cells that has some type of issue associated with it. So we have our helper T cells that are going to coordinate the immune response and will also help to stimulate our inactive cytotoxic T cells by a process known as co-stimulation. We'll have our memory helper T cells and our memory cytotoxic T cells that will stick around and wait for that same antigen to come back in so that they can re-undergo clonal selection and produce the army of cytotoxic and helper T cells in response to that antigen. And we'll have the cytotoxic T cells that will actually go out themselves to our infected body cells and insert perforins into their cell membrane that leads to the demise of the infected body cell. This slide will show you the activity of our cytotoxic T cells. Once again, you can see the CD8 proteins on the surface of our cytotoxic T cells bound to the MHC1 proteins on the surface of our infected body cells. You can see the microbes inside of the infected body cells over here. Cytotoxic T cells are eventually going to go out and bind to these infected body cells, and they're going to release perforins, but also release substances known as granzymes. We've already discussed how perforins work. Perforins, you can see, will poke into the cell membrane and form a pore. Granulysin is often associated with perforin. Granulysin specifically will make its way through the pore that's been formed by the perforin and digest all of the inner components of the infected body cell. This destroys both the body cell and the microbes within it. This is one action of our cytotoxic T cells, but we also have granzymes that can be released at infected body cells also. Granzymes stimulate a process known as apoptosis, which is programmed cell death. So when these substances are released to our infected body cells, that's going to signal to our own cell to destroy itself. This will then lead to the release of the microbes inside of that infected body cell, ridding them of their protection. And once they are exposed, phagocytes can come around and eventually destroy those microbes that were initially infecting our body cell. Once our cytotoxic T cell has dealt with the infected body cell, it'll detach and eventually go around looking for the next infected body cell. So our cell-mediated immunity involves T cells. We have helper T cells, cytotoxic T cells, and memory T cells. When cytotoxic and helper T cells are activated, they're going to undergo the process of clonal selection. This produces many more clones of these cells and also produces memory cells that will be present the next time that pathogen tries to make its way into our body. So we must remember, with the cell-mediated immunity, antigen presentation is a must. We cannot activate our cytotoxic or helper T cells without the help of an antigen-presenting cell. These concepts conclude the cell-mediated immunity.